Dynamics of Marek's Disease. A presentation by Professor Gershom Zaitchik, Faculty of Medicine, Hebrew University of Jerusalem. Marek's disease is a disease of domestic chicken caused by a herpes virus. Birds get infected by inhalation of infected dust which contains the virus. Marek's disease occurs at 3 to 4 weeks of age or older and is most pronounced or common between 12 and 30 weeks of age. It proceeds through two stages, acute and chronic. Some symptoms or syndromes of this disease: lymphomatosis, neuropathy, brain pathology, iridocyclitis, and lymphoma. Marek disease is a multi-dimensional dynamic process driven by herpes virus. Symptoms are the response of the organism to the virus. And here you have a description: Marek disease, a model for herpes virus oncology. I use there figure 1. It's modified drastically, but it shows the disease progression. So the disease starts on day 6 something, and then you see that there's a viremia due to lysis of cells, B lymphocytes. So lysis is virus-triggered lymphocyte death manifested by immune suppression. So since lymphocytes are dying, immunity is suppressed, and after some 2 or 3 weeks, you get another phase. The disease enters its chronic phase, and it is manifested by latency, again lysis, immune suppression, and finally, at the end of the whole chronic phase, you get a lymphoma. So this is published in figure 1. So in order to understand the dynamics of Marek's disease, let's study the interaction between a virus and a cell. Interaction between cell and virus has two outcomes. Lysis, when virus hijacks cell genes in order to produce virions, and the cell dies. Lysogeny, when cell hijacks virus genes in order to protect itself from it and from other viruses, and the cell lives. So here you have a general cycle of a virus, and so virus infects a cell, and its genes are now incorporated into the genome, and the cell starts producing virions. And as the virions accumulate, the cell dies, which is called lysis. The virions enter the neighborhood, infect other cells, and so you have a cycle of lysis, which is production of viruses. But occasionally the same cell may hijack or incorporate viral genes for protecting itself from other viruses. So it's not a lysis, it's called lysogeny, and the viral genes in this genome prevent similar viruses to enter the cell. So it's a kind of protection, and this protection is inherited by the clone of these infected or lysogenic cells. Now, the same happens in Marek disease. The Marek disease virus is tropic to B lymphocytes and epithelia. During acute infection, most virions are eliminated by the immune system during the lytic infection. Some are incorporated into cell genome, which are lysogeny. This phenomenon is also called latency. So here you have the same cycle, only what we know now that once the viruses enter the extracellular fluid, they are eliminated by the immune system. And but the phase is called lysis. Again, it has several names. Latency, it's called the state lysogeny, and the genes in the genome are called proviruses. The viral genes in the genomes are called proviruses. First stage. So day 6 you start viral production, which is accompanied by lysis, cell die, and then later on the disease progresses into its chronic phase, where you have more and more lysogeny, and it is called latency, which means more and more cells protect themselves by incorporating viral cells. Like here, again, day 7 is here, and the chronic phase is here, and this explains latency. But what we see here also in Marek's disease, that parallel to latency, we have again another bout of lysis. So here you had viremia due to infection, but here the lysis is due to another mechanism, which means that the genes which protected the cell against other viruses are activated by stress, and once they are activated by stress, they produce virions, and you have a second lytic phase. And here you have the scheme. We started with infection during the acute phase, and we had this lytic cycle. But in the chronic phase, stress activates the genes, and they also start, or the cells start producing virions, and you have this lysis. Now, where does all this happen? Here we have the lymphocyte lineage. You have the blood stem cell, lymphoid stem cell, lymphoblast, and what we see here, that we have a lineage which is differentiating, and we are talking only about lysis of B lymphocytes. The virus is less tropic to T lymphocyte or to natural killer cells, and therefore when we speak on lysis, it's a lysis of B lymphocyte. And here you have the scheme vertically, that this stress hit the activated lysis in a lysogenic cell. Where does it happen? So let's remember that the stem cell remembers the lysogeny, and here are all its differentiating lymphoblasts. But since they are differentiating and since they are short-lived, sooner or later they will die. But the information about lysogeny is generally in the stem cell. Back to our scheme, so we have lysis in acute phase. Since we have depletion of lymphocytes, we have immune suppression. We have lysis in the chronic phase, which is due to activation of genes. Again, lysis and immune suppression. And now you have to remember that we are talking that the whole thing is happening here. Latency accumulation happens here, and lysis happens here. Finally, we got to the stage of lymphoma. So we started with acute inflammation, which is called lymphocytosis. Then we got chronic inflammation. And finally, within the chronic inflammation, a lymphoma emerged. So note that lymphoma is the latest stage in a chronic infection. We never get a lymphoma in acute in the acute state, and even in the subacute state we don't. Lymphoma appears as in the last stage, and this is very significant because it happens in all cancers. So we saw lymphoma emerges within a chronic inflammation. This happens also to sarcoma, like I shall show. Sarcoma emerges also within a chronic inflammation, and I discussed it in the presentation Clinics of Avian Leukosis Disease. So Rau's sarcoma appears in the last stage of this leukosis. Hepatoma, the same thing. You start with acute hepatitis, you get subacute hepatitis, smoldering hepatitis, cirrhosis, and finally you get hepatoma. So there's a general pattern which I call law. Any tumor emerges within a chronic inflammation, and this chronic inflammation I called cancer field. It is described here, virus-induced cancer and the cancer field. So we are interested to cure Marek's lymphoma. Obviously, if you want to cure Marek's lymphoma, first of all cure Marek's disease, because if there's no disease, there's no lymphoma. But there's another interesting phenomenon which we are going to discuss now, that if you infect the chicken with turkey herpes virus, you can cure its lymphoma. So here you have, first of all, a paper on the epidemiology of a herpes virus of turkeys, possible sources and spread of infections in turkeys' flocks. So essentially, the infection and the epidemiology of turkey's virus is the same like the Marek's lymphoma, as we shall see. They write, the present study was designed to investigate natural infection of commercial turkey flocks within herpes turkey virus, focusing on the origin of infection, its spread and its persistence within the flock, and its pathogenesis. So, as I said, that it's the same story also for the Marek disease virus. Some of the important epidemiological features of natural herpes turkey virus infection in turkey flocks revealed in this study include early appearance of infection, its rapid spread throughout the flock, and its persistence. And the same happens in Marek's disease, and it's very significant. So if you take the figure 2, you see that the virus appears on the day, on the third week, and then you get rapid virus proliferation, and this is the lysis phase, and you get viremia in 100% of the turkeys. And you can prove it with antibody, which is proportional to the virus load. But what is significant about this figure is there was no indication in this study that herpes virus in turkey induced disease in turkeys. So all these turkeys were clinically healthy, but they were viremic. It's a very important observation. So how do we prevent Marek's disease? Here you have a review. Marek's disease is a highly infectious neoplastic condition of chicken caused by herpes virus. The virus is cell-associated in tumors and in all organs except in the feather follicle where enveloped infectious virion egress from the body. So the non-pathogenic herpes virus of turkeys is ubiquitous in turkeys and is probably spread horizontally by the airborne route. When chicken are inoculated with this virus, they don't get Marek disease. The herpes turkey vaccine is safe and highly effective in preventing Marek disease under field conditions, and most chickens throughout the world are vaccinated with this vaccine. So what does it all mean? We have two viruses. One is MDV virus and one is HVT virus. They are relative. You can regard them as cousins. They have a similar genome structure and similar envelope structure. Only this one is pathogenic and this one is not pathogenic. Both create lysis and viremia. This is the essence of Marek disease, but this one causes a viremia and is clinically healthy. So one step further is, if we treat a cell, a lymphoblast, with herpes turkey virus, he will get lysis and viremia, but he will not get any other symptoms. Now, if we try to infect him again with MD virus, the virus will be blocked and will not enter the cell, and the cell will not produce MD viruses. So here you have, again, infect the cell with MD virus, you get lysis and viremia with MD viruses. Infect the cell with turkey virus, you get turkey virus viremia, but without the clinical symptoms of this Marek disease. And this is the essence of interference. The HVT interferes with the MDV, and so you can cure Marek's disease because they interfere. So this phenomenon of interference was discovered also in other chicken. In 1911, Francis Rous discovered the Rous sarcoma virus, RSV, which causes chicken sarcoma. In 1960, Harry Rubin discovered a virus which prevents Rous sarcoma virus from causing a chicken sarcoma. He called it the resistance-inducing factor, but I, in his honor, call it a Rubin virus. So Rubin virus has a similar structure, genetic structure like Rous sarcoma virus. It has a similar envelope structure, protein structure, but it is not pathogenic. What do I mean pathogenic? You get leukocytosis, but you don't get Rous sarcoma virus. And so what I describe here, that Rubin viruses cures Rous sarcoma here in this presentation. In other presentation, I discuss viral interference, how to cure Rous sarcoma, clinics of avian leukosis disease, and how to cure chronic hepatitis. All these are based on interference between viruses. So here the scheme of virus interference. Infection with a non-pathogenic virus blocks the infection of a pathogenic. And I call these non-pathogenic viruses in honor of Rubin, Harry Rubin, I call them Rubin viruses. Rubin viruses, it has the same gene, similar gene structure like the pathogenic virus, and the Rubin virus does not cause any tumor. So if you infect a cell with a Rubin virus, it protects it against a pathogenic virus, and this is the essence of viral interference. Now, what we can learn from the current medical dogma? Current medical dogma does not believe in this, what I told you, and they say that cancer is caused by broken mutated genes, and viruses only enhance the break of the genes. And you can read a review, The Cancer Genome. So what do they write? The mutations found in a cancer cell genome have accumulated over the lifetime of the cancer patient. Some were acquired when ancestors of the cancer cell were biologically normal, showing no phenotypic characteristic of a cancer cell. We shall see immediately figure 1 what they mean. DNA in normal cells is continuously damaged by mutagens of both internal and external origin. So let's look at figure 1. It is a summary of current medical dogma, what is cancer about. So here you have a fertilized egg, and as we grow, we accumulate mutation. Initially there are 2 or 3 mutations per cell, not so bad, but then we cross a threshold and we have more and more mutation, like here early invasive cancer, late invasive cancer, and even chemotherapy causes mutation. So this is what happens to any one of us. And yellow are intrinsic mutations, blue are external mutation. Then we have the mutator phenotype is a tumor with a lot of mutation which drive it, and the more mutation it has, the faster it grows. And among these mutations you have passenger mutations, you have driver mutation, and you have chemotherapy mutation, which means passenger mutation drive tumor, and they compete. There are different clones, and the more passenger mutation you have, the worse the prognosis. So now let's apply the logic to Marek's lymphoma. We just saw it. We know what happened there. So again, according to the current dogma, you have accumulation of mutation exactly like any other tumor. But what does it mean? You can have any mutation which you wish in this lymphoma. Once you treat the chicken with herpes turkey virus, lymphoma regresses. So in our story here, Marek lymphoma accumulates mutations. So what? It may accumulate mutation. We treat it with a Rubin virus, here herpes virus, and the herpes turkey virus, and lymphoma disappeared. So this is the major message of this presentation, and this is the message of Marek's lymphoma. It somehow does not obey to the current dogma of the mutation metaphor. Next, you are invited to listen to mononucleosis, which protects against lymphoma, and mononucleosis is also caused by a herpes virus. Enjoy.